Okay, so today I'm going to be talking about my journey in science, breaking stereotypes, and my most complete body of work where I've been using machine learning to find stereotypical characteristics of metastatic melanoma. So I've been doing research for a while. Oh, my computer froze. Okay, I've been doing research for a while. My early days of research started in my front yard. I was, always seen out there looking for insects, playing with insects, and just trying to catch butterflies. And that was my favorite thing to do. And my mom would take me to the local library so that way I could check out books and learn more about bugs. And through my investigation of bugs, I was able to find my first exposure of a scientist. And I learned that an entomologist is someone who studies insects as a career. And from that point on, I wanted to be a scientist. I was hooked. And when people would ask me, Andres, what do you want to be when you grow up? I would say, an entomologist. And they would look at me and have a very confused look. And their confusion was twofold. One, not very many people in my community knew what an entomologist was or is, and two, a young, poor kid from Central California, Fresno, California, largely being migrant Latin workers. I did not fit that mold of what a scientist was. They had this particular um, stereotype of a scientist, and I just didn't fit that. Now you might be thinking, oh, maybe people from different communities aren't exposed to different scientists, but these were educators. These were my teachers. These were my principals. And I think now, currently in this climate, we're seeing more and more evidence where people are discriminatory based off of stereotypes. And we're seeing this more widely. And that's something that I had experienced very early on. And I think a lot of URMs have. So that didn't really stop my interest in science, and it stopped me pursuing that. And I knew once I got to university, I would be able to have my first real shot to be an academic researcher, to be a scientist. And I was so excited. And I got into California State, Fresno, and during my second week, I was emailing professors trying to find a research lab, and I was lucky enough to find one. And within a couple of days, I was already in the lab. And so this is me at my first conference, wide-eyed, bushy-haired, nervous as heck, but proud to be presenting research. And I was very, very fortunate. I was fortunate, and especially given this is a DEI talk, I want to talk about how programs helped me. One, I had an amazing mentor. We had a great personal relationship. We would listen to music in the lab. We would talk. I got great hands-on experience from him, and it was an amazing time. And I really enjoyed researching in that lab. And I was so happy that he put the amount of time and he had an interest in my development. And that's something that's key for URM scientists. And when I mentor students now, I tell them focus on the mentor. That's the most important thing. Science is second. And for me, remember, I told you I was interested in entomology. I wanted to be an entomologist, but this lab was a molecular biology lab, and I was using computational models in molecular biology, and had nothing to do with what I wanted to do. But I had an open mind, and I just wanted to research. I wanted to learn. And so, one of the other things that helped me during my academic career at Fresno State was to be funded by diversity initiatives. So LSAMP, McNair, Metro are programs that I was fortunate enough to be funded through. And what's important to note is that a lot of URMs have to choose between working and doing research, because typically research is unpaid, but there are these initiatives that do pay students. However, sometimes they can be very limited in the spaces that they're offered. So for the faculty out there and for people that are able to do something about this, one, pay your undergrads. Two, increase the diversity of your undergrads. Take URMs. Change the face of your labs to be more inclusive. For the people out there that are able to make changes, and hopefully I'll be talking to you pretty soon, to increase the amount of funding for undergraduate researchers in URM communities. Sometimes it's on the order of dozens. We want to expand this on the order of hundreds to make sure that there are a lot of students getting research opportunities. And as Gabriella said, that is extremely pivotal for first generation URMs to be able to get it into grad school. For me, I knew based off of my parents' experiences and based off of my early experiences, I had to be 2 times better than my non-URM competition in order just to be seen as not as a token, but as a quality candidate. And so I tried my hardest to do research for 4 years. I did a summer internship, and I was lucky enough to get a summer internship every summer. And it was because of these programs that taught me how to write an abstract, how to write a statement of purpose, which programs were open, and how to do all of these things allowed me and gave me the support and the experience and the knowledge to do so because my parents didn't know how to do so. And that was extremely pivotal. But in order for us to not just increase diversity and have the equality, we need to create a space and a support community for young URM scientists. And I'm from Fresno State, and this is a Hispanic serving institution. But I wanted to give back. SACNAS is a national community of URM scientists. And I wanted to bring SACNAS to Fresno State so that way they were connected to the national organization. So this is me here. This is my cousin. This is the President Castro, Lilia de la Cerda, the director of LSAMP. And he had pledged monetary funds. He had pledged support for many, many years. And SACNAS has been thriving at Fresno State. And the important thing is that these two individuals were SACNAS members from their early scientific careers. They understood the importance and they understood the impact that this would bring to their school. Now, many of you may not know, but I'm a transfer student. I started part of my PhD work at UT Southwestern Medical Center. And when I got into UT Southwestern Med Center in Dallas, there weren't a lot of URMs. There was not a strong URM community. I felt alone. I wanted to get to know people like me. So created another SACNAS chapter. I've created community when there is no community. And I was so fortunate to work with these individuals and create this community and to be able to do outreach for students in Dallas. It's a highly, large Latin population. We did outreach to students to show them that there are scientists like them, that this is a real possibility. And from our given, spectrum of experiences, some of us, I didn't have best GRE scores or best GPAs, but to keep working at it, and that there are opportunities for you, and there is a place for you in research, despite not seeing scientists as a diverse population. So I had to transfer, and I transferred to UCSD, and I was nervous, but I felt some comfort in that there was a SACNAS chapter. So I got to UCSD and I started looking up the SACNAS chapter and emailing, and then I came to realize that the SACNAS chapter was no longer active anymore. And I came straight into a lab. There's no diverse individuals in my lab. I didn't know of any diverse individuals on campus. There was no highlighted page on the websites. There was no DASL seminars that I could find people like me. So what did I do again? Started with help of many individuals, another SACNAS chapter. And what was different about this chapter is that we're a mix of undergrad graduate students. So at Fresno State, it was undergrad only. At UT Southwestern Med Center, it was graduate only. This gave us a unique opportunity for graduate students and postdocs to mentor students and to help them with their applications, but also to advocate and to help these students find paid research positions, and for faculty and graduate students to work together to make these proposals so that way we can get more increased funding for undergraduate researchers in the URM community. And that's something we'll actively doing. And you'll probably be getting emails from me in the next couple of days. And so with that, having that community, creating equality, and increasing our diversity initiatives is key to change the representation and to, like Gabriela said, get people in our communities to trust us. Now, given all of that, there's something that I continue to face and a lot of URMs continue to face every day, and that is discrimination. Even here at every institution, ever since I was younger, I faced discrimination based off of stereotypes. I spent a majority of my time researching, and I was excited when I was going to graduate interviews and interviews at multiple places. And there was one particular interview. I went in, I had my name tag on, had my name, had Fresno State, and I walk into the office with this PI, and they immediately said, "Why the bleep do I care about you? You're from a bleeping no-name school." And I was floored. I didn't know how to handle that. I'm a young student. I'm just excited to have been given the opportunity to interview, and this happens. And so this person opened up my file and then said, "Oh, you did research at XYZ institutions in the Bay Area in Southern California. You've done research for 4 years." Okay, that makes sense. Because immediately, based off of where I'm from, how I look, I did not fit that mold of this person who is a quality applicant for that university. And let me tell you, that is not something that's unique to that university. That's something that there's a community of us that we share these experiences, and it happens across the nation. And sometimes it happens, and this is what I want you to think about, to have questions so that way when we have our open-ended discussion, what do we do? This happens in admissions committees. This happens in other committee meetings. What can you do as faculty? How do you speak up? When this happens to you as a student or a postdoc, how do you be your own advocate when sometimes there is no advocate for you? How do you find a community to help you? And what are possible repercussions and consequences for those individuals that choose to discriminate and choose to do these terrible things to URMs? What are these possible repercussions for them? Because now this may make you feel uncomfortable. There are systems in place to silence, to hush, to make those complaints go away. And I tell you from firsthand experience that these things exist at universities. And there has to be things we can do to change. You are all here, you are invested in DEI, you want to make a change. So think about this as I go through the next portion of my talk, so that way we can have a conversation. And for those of you in the audience that may have felt these things, I'm here. I'm here. Sockness is here for you. Doesn't matter who you are, come to us and we'll be that support system for you. So I want to go into the next part of my talk. This is my most complete body of work, researching metastatic melanoma and using machine learning to find cool characteristics and features. So features are just characteristics of a cell. So one of the things is that melanoma has the highest mutational burden of any cancer. And this poses a real challenge for finding a genetic signature for metastasis. And not only does it have the highest mutational burden, but melanoma is the poster child of metastasis. And 1% of the primary tumor metastasizes. But what is special about this rare cell population? You have this patient, this is a classic thing, and this has been reproduced in multiple other papers, but this is a classic panel for this patient. The patient had a primary tumor in the arm, and it metastasized, meaning it spread. That 1% of cells spread to dozens of spots on his body and multiple organs. And with targeted therapy, it seemed like there was complete remission. It was an amazing turnaround. But we all know the story. There's remission. Cancer comes back. And there's some resistance to that therapy. So it intrigued me of what gives this 1% of that primary tumor the right. Why are they special? Why is it that they can do such complex tasks, such as surviving in multiple environments, being exposed to different stressors, and still survive and thrive in the host? When I was at UT Southwestern, I was lucky enough to be able to have access to a repository of metastatic melanoma biopsies and access to a mouse model. So what they did, this Morrison group at UT Southwestern was our collaborator. What they did is they took stage 3 melanoma biopsies. They transplanted them into mice, and they found a metastatic efficiency. What does that mean? Metastatic efficiency is the degree of which it invaded throughout the mouse. And then they correlated this outcome in the mouse with the outcome post-mortem of the patients. And they found it was a nearly complete, correlation between these two. But by doing this, they revealed that there was actually a binary classification of metastasis. So, metastasis is very deadly for melanoma patients. It's poor prognosis. But what they didn't know was that there was actually a stratification of these. And the stratification is this. You have low efficiency metastatic melanoma. So this is a mouse. The mouse has been engineered with luciferase, and I add luciferin in, and there's a bioluminescence reaction. And this is showing the primary tumor on the right side of the animal. And then here we have the dissected organs, and we're seeing that this is a metastatic microtumor in the lungs. And so for low efficiency, it's metastasis to the lungs and lymph nodes, which is seen in the patients and seen in the lungs in the mice. Now, high efficiency is classified as metastatic tumors in multiple organs, but also as large tumors, similar to what you saw in that patient. And it is that these where we see that there is really truly poor prognosis. And so what the idea was to do, and what many other groups have done since, is to take all of those, metastatic tumors from low and high and to grind them up and sequence them. And so that's what we did. We did some whole exome sequencing. We annotated the sequences based off of, oncogene or likely oncogene using a database for melanoma. And we found that mutations fail to discriminate metastasis, either qualitatively, the presence or absence, let's say, of just gene A, gene B, or quantitatively to show a spectrum of mutations for each patient. And this was done quantitatively with the Jaccard similarity distance. Now, I'm going to show you a confusion matrix, and I'll explain what that is. So let's say you have 498. 498, when it's paired with itself, they're identical. So you have a blank white space. Now, the heat map over here for the Jaccard distance is the colder it is, the more similar it is. The hotter it is, the more dissimilar these two tumors are. So what we saw is, in general, there was no striking pattern between low and high metastatic efficiency based off of the mutations in the somatic mutations. So the next thing to do is, okay, maybe we could try a functional assay. Maybe this will tell us more allow us to discriminate between high and low metastatic cells. So we use standard cell biology assays, extracellular acidification, oxygen consumption, proliferation rate. These are functional assays that we hoped would parse out the differences within these two within this rare population. And we were not able to do so reliably and robustly. So the next thing we thought of is, can we predict metastatic efficiency simply by looking at the cells, and do so in a label-free manner? So let's take some images, some movies of cells, and let's see if we can just, by looking at them, tell a difference. And this is important too, because if you get a biopsy is very precious. If we can do so with a low number of cells, a low amount of that biopsy, and where we don't introduce any markers, don't infect, nothing, so we preserve that state, and we image them on a matrix that's similar to what their microenvironment is. And can we see any differences between these two populations in the way that they look and the way that they move? And so one of the first things to do, and many groups have done, is just using shape. You just look at the cell and see the shape. And shape can mean eccentricity, which is just the aspect ratio, area, perimeter, convex hull. These are just quantitative terms for quantifying cell shape. We used all of them, and to see if can we discriminate between high and low metastatic efficiency. And the answer was no. We built a classifier based off of static shapes, and we could not classify accurately. We did using the shapes, but time average. Remember, I have movies. We did a time average of those shapes over a given amount of time. Still, no accurate classification. We tried a bag of words algorithm, which essentially is if you have a bag of sentences broken up and there was a word that occurs most commonly, then maybe that will be a standout feature, a standout word, and in this case, a standout shape that possibly could lead us to a stereotypical shape that discriminates between high and low metastatic cells. That did not work either. So, machine learning is big, right? A lot of people are using machine learning AI, and it's doing a great job at classifying and being able to discriminate between populations, and it's very good at that. But one of the things that it's not good at is allowing us as research to see what are those features, what are those characteristics of the cell that allow us to discriminate between these populations. So we wanted to know what those features were, what those characteristics were, because that allows us to then backtrack and see how do those features connect on a molecular level to the things that we're seeing. And that may point us in a direction where genetics and functional assays couldn't help us find that key feature, that key thing that differentiates these two populations. But maybe finding a shape feature or the way a cell looks and be able to backtrack and find something molecularly that could tell the difference between these two populations. So we used unsupervised machine learning. So you take an input image, you extract features, you have it in this 56-dimensional feature set, and then we want to decode that because remember we want to see what they look like. So we decode that feature set and we reconstruct an image based off of that 56-dimensional feature set. And then we could see what it is that the machine is learning about the cell. And let's say, okay, we have this. Now let's see, is this enough to allow us to discriminate between metastatic cells? And we see that these are the autoencoder. This is the unsupervised machine learning based features that they do significantly better than these shape features. And we were able to discriminate between metastatic cells. And that was great. But still, it's not all the way of what we wanted. We want to know what is the feature that allows us to discriminate. Is there one? Is there multiple? What is it? So to find that, we took that 56-dimensional feature set, and we had this adversarial autoencoder, classifier that correlates confidence scoring, some weighting, in order to identify which of those 56 features are the key features. And luckily, we found that feature 56 allows for discrimination between high and low metastatic efficiency. And that was great. We were lucky that it was just one, and we found this feature. But what is feature 56? So this is, I'm going to explain how we found what feature 56 is. So this is a reconstructed image based off of those 56 dimensional feature data set. Okay, so this is the reconstructed image. Then we're looking at the difference between the raw image and the reconstructed image. And we're seeing how enriched is feature 56. And then what we're seeing in these cells, and the difference of this is a difference in light scattering or refractive index, as well as subtle pseudopodial extensions. And so based off of a generative model in machine learning, we were able to identify subtle features that we could not see, and that were changes in the refractive index and slight pseudopodial extensions as a functional hallmark of highly metastatic melanoma. And now this allows us, and this kind of connects into my work in the HAL lab, of what I'm doing to investigate how those functional hallmarks, how they relate to a cellular molecular context. And to find out more about this and to read more about the methods, it's really cool. Check out our preprint and bioarchives, and let me know if you have any questions. I want to thank the HAL lab. I didn't get to talk too much about the research I'm doing there, but definitely thank them as my new home here at UC San Diego and having a very supportive mentor and an awesome lab to work with. And I want to thank the Saritsky lab at Ben Gurion University where I was one of my mentors, direct mentors on the project at UT Southwestern, and maintain a great relationship and awesome collaborator to work with. And I want to highlight SACNAS. So you are all here because you're interested in diversity, equity, and inclusion. Please sign up. Please follow us on social media. We need faculty advisors. We need faculty members. We need postdocs, graduate students, undergrads. So please tell your friends. Please get involved. We really want to expand this so that way this is a community for everybody, and it's a community to support those of us in the URM community. And thank you.
All right. Thanks, Andres, so much for sharing all your work, and both in research and in SACNAS and building all these wonderful communities. We've got a little bit behind, so if some people have to leave, we're really glad you're able to join us for DASL today. But now I think we can move on to the open-ended question and answer session. So if you have any questions for Andres or Gabriela, please either go for it or enter it in the chat if things get a little rowdy. But again, thanks to both of our speakers today for being here with us.
I have a question.
Yes, please go ahead.
Oh, this is for Andres. You said subtle pseudopodal extensions. So those are like the cells, reaching out. Is that for, you think, for metastasis between cells of the blood vessels, or to get into the blood vessels, or perhaps that's just too much speculation?
No, so we actually have continued with this in a 3D matrix and with some, cellular architecture. And what we're seeing is that sometimes those arms are releasing exosomes that contain things that can degrade, like MMPs that could degrade the matrix around them, as well as sometimes they're using, as you saw in the video I had of the different cells, there's some cells that are moving, grabbing the collagen and pulling themselves around. And sometimes as well, they catch another cell and they kind of become a leader cell and they start walking with them. It's really crazy how these cells use their pseudopods in order to do vastly different things that can enable them to be more metastatic.
Really interesting. Thank you.
Thank you.
I think Dr. Neal had a question. Would you like to share?
Sure. Gabriela, you mentioned you were a first-gen college student. So what led you to pursue science in the first place?
That's a good question. So yeah, science wasn't in my upbringing at all. Everything was very magical and very mystical. So I think I kind of had I felt a bit uneasy about that. I was always interested in science, and in high school, I had a really great teacher. I think something that's really important for first-gen students is having really great mentors and really great teachers. And I had a really great biology teacher that, gave me an opportunity in a research lab in a university when I was in high school. And I thought that was insane. And I went for it and I loved it, and I'm here today. So that's what it is.
All right, please go ahead and ask any questions in the chat. I can ask Andres. I think it's so amazing that you formed all these SACNAS chapters, and I'm just wondering, how did that, what's the secret? What's the trick to building these communities, these different institutions, not just in one place or at one time, but multiple times?
You have to be a little bit crazy, I guess. It's hard, especially trying to continue with research and go to conferences and stay active in classes. It's definitely hard, and I think that's something that a lot of the founding members at every institution have found out, that it's hard. But I guess the secret is just working with people. working with them and getting them to create a community and leaning on each other, making sure that in the first founding chapters and the first founding members, there's roles, but really we're all doing this together. We all work with each other. There is no one better than, or there's no titles. It's just we all need to work together to make this community a real community and to be thriving. And once you start thinking about, well, I'm this position and that position, and I'm doing X, Y, Z more, it breaks down because now you're doing it for yourself. So you really have to be altruistic in the message, in the mission, and for it to be not about you, but for those behind you that are coming up and to create a better place for them. And honestly, the reason I do it is for the little me. He didn't have anybody, he didn't have that community. And there are other little me's out there. I have a young daughter now, and I want to make sure that though there are communities out there and there are things in place to show her that she's allowed to be anywhere she deserves to be and she wants to be, and that there are going to be people there to mentor her and to help her along the way.
Wow. Inspiring. Dr. Neal, do you want to ask your next question?
Initiatives, yes.
Oh, there you go.
So right now we are working with the transfer students and high school students. We've done a lot of outreach for them. And so that way we get them interested in STEM, high school students just interested in STEM at any level. And so even if some people can't go to college, but we want them to get them interested in STEM regardless. It provides a great career opportunities, flexibility, and it's just fun. So we expose them to that. And then we want to make it clear that while we do want to increase diversity at the faculty level, there are different career paths, and you don't have to go into academia. We just want you in STEM to change the face of STEM. And then for the transfer students that are interested in UCSD, we want them to get involved in research. A lot of them are interested in graduate school, so we're partnering with faculty to get them into research labs. We're partnering with Dean Antony to make sure that they get money and they get funding for this. So one of the things that we're currently doing, is trying to get every department, every dean, and everybody to sign this petition to show that they're pledging X amount of dollars, they're pledging X amount of support. So that way we can then take to our chancellor and see that the large support from all the different departments in STEM, and that way there's a big university push to make sure that SACNAS is a central resource and a central community for everyone, and to provide undergraduate research positions and mentoring in that capacity. And then they have access to a national conference that they're able to go to. And that's what's going to be really helpful. Like Gabriella, she said she went to these conferences. I went to these conferences. That was awesome. That was pivotal. I got to present at a national conference and get questions from outstanding faculty members and scientists. That was great. So we need to increase that. We live in San Diego. We don't have the Latin representation at UCSD. We need to do better.
I had another question for Gabriella. I'm curious if you think that having that kind of background of, maybe not being exposed to scientists, or, having this, different relationship with just kind of more mystical aspects, do you think that kind of like changes how you do research, or like what research questions you think are important? I guess I wonder, if you feel like you've fully integrated as a scientist, or whether you still have some unique perspective because of that.
Yeah, definitely. I think early on in my science career, I was still on like the bridge between this world of these beliefs and science, and I was just trying to make that bridge. I did a lot of thinking of, I'm going to try to go into nutritional supplements and see how to cure diseases with, nutrition. And I think this is great. And I think that we need to do things like this But then, I got carried away with the biomedical sciences and the drugs, and my family kind of looks at me like, oh, why are you studying drugs? But I think that once we understand that these mechanisms and we can find like natural products or something that can help us other than these drugs to get more trust in the community, that's probably, I think, the goal. But yeah, I don't know if I've fully integrated. I'm doing my best.
I have a comment and then a question. The comment is, I don't know that we need to like all ascribe to a super hardline sciencey thing. I think you would be shocked actually at how many people in the biology department read tarot. There's a lot of us. So the question is this: I am a white lady. How can I be a better ally? How can I simultaneously boost voices of URMs and first gen students, and also not make them do all the work, right?
I think you're doing a great job, Hannah. I'm not an expert in this. I think that, having a platform like DASL to have us talk about our research and our experiences is great. I think, participating in these and just listening is also good. But I don't know, maybe Andres has something to say.
Yeah, no, I do think Hannah's doing a great job, and it would be great to hear, Hannah, how you do that, how you kind of do very well manage that line of being very supportive but not like taking over these certain things. So I think that's awesome, and it would be great to hear from you. One thing that I always, for allies, that they ask me, how can I get more involved or how can I help and everything, because I feel uncomfortable when I go into a room and there's not a lot of people that look like me, and I feel like I'm not supposed to be there. I tell them, get used to feeling uncomfortable and reflect on your actions, get used to being uncomfortable. I'm uncomfortable all the time, and it's okay to be uncomfortable. I'll be uncomfortable with you, and we can talk about things and learn how you can be a better ally, how I can help communicate my experiences better, and for you to understand that sometimes maybe I deal with my experiences differently than others, and it may come off too jokey or edgy, but that's just a way that I deal with my experiences. And so we have to be accepting of all ways people deal with this trauma. So that would just be some